Have you ever just stopped and wondered, how do you become you? I mean, really. The answer is written in this incredible microscopic code, a set of instructions tucked away inside nearly every single cell in your body. So today, we're going to unpack that amazing molecular journey that turns a simple code into, well, the complex, living, breathing person that you are.
Okay, get this. Every one of your trillions of cells holds about 2 meters of DNA. That's right, 2 meters. That's taller than most people. And all of that is crammed into a space so tiny you can't even see it without a super powerful microscope. So the first question is, how on earth does the cell store this massive library? And the second, even bigger question is, how does it actually read these instructions to build everything from your eyeballs to your immune system?
Well, the answer to that is beautifully laid out in what biologists call the central dogma. It sounds complicated, but it's really the master plan for all of life. It basically says that information flows in one main direction, from the permanent storage, which is your DNA, to a temporary message called RNA, and then finally into the things that do all the work, the proteins. This three-step flow is the exact process we're going to explore.
All right, so let's start at the very beginning, with the master blueprint itself, the DNA. You got to understand, this molecule is like the cell's crown jewels. It's the complete, unabridged instruction manual for life, and it's kept under lock and key inside the cell's command center, the nucleus.
Just think about that number again for a second. 2 meters of DNA inside a nucleus that's only 6 micrometers wide? That's like trying to stuff 40 kilometers of superfine thread into a tennis ball. I mean, it's just a mind-boggling feat of engineering, and it turns out the cell has a brilliant way to do it.
So here's the solution, an amazing multi-level packaging system. First, the DNA wraps around these little proteins called histones, which makes these structures that look kind of like beads on a string. But it doesn't stop there. These then coil up even more, and then loop and fold and keep condensing until you get a chromatid, and you've compacted it over 10,000-fold. That's how it's even possible to store it all.
Okay, so the library is all neatly packed away. But what happens when a cell needs to divide? Well, it has to make a perfect, flawless copy of its entire DNA library, all 6 billion letters of it, for that new cell. And this process is called replication. And right away, we run into our first huge challenge. I mean, copying 6 billion letters is an absolutely massive job. And a single mistake, just one typo, could be a total disaster for the new cell. So this whole process has to be not only incredibly fast, but unbelievably accurate. And of course, the cell has this sophisticated molecular machine to pull it off.
And that machine works in 3 basic stages. First up is initiation. That's where the machinery finds the exact right place to start. Second is elongation. This is the main event, where the new DNA strands are actually built. And finally, termination, where the whole process gets wrapped up neatly. But, there's a catch. The 2 strands of the DNA double helix actually run in opposite directions. It's like a two-way street, but the copying machine can only go in one direction. So what does the cell do? It uses 2 different strategies at the same time. One strand, the leading strand, gets copied in one smooth, continuous line. But the other one, the lagging strand, has to be copied backwards in these little chunks that get stitched together later. It's just a brilliant workaround to a really tricky chemical problem.
So, we've seen how the master blueprint gets copied for new cells, but what about using that information in a cell that's already up and running? We need a way to get those instructions from the protected DNA library out to the cell's little factories.
And that brings us to challenge number 2. The DNA, remember, it's way too precious to leave the safety of the nucleus. Taking that whole master blueprint out into the rough and tumble environment of the cell would be like taking the original Mona Lisa on a camping trip. You just can't risk it. So the cell has to make a working copy.
And that process of making a temporary copy is called transcription. It's like photocopying just one page out of a giant encyclopedia. A specific gene, a single instruction, is rewritten into a new molecule. This mobile copy is called messenger RNA, or just mRNA. It has the exact same information, but it's in a form that's totally disposable and, crucially, can leave the nucleus.
Now, DNA and RNA are really similar, but they have a few key differences. DNA is that stable, double-stranded master blueprint that uses the letter T. RNA, on the other hand, is the single-stranded working copy, and it uses a slightly different letter, U instead of T. These features make it the absolute perfect temporary messenger.
So now we have our mRNA message. It's made it out to the factory floor.
a little structure called a ribosome. But we have one last major hurdle to clear. We have to translate this message into a completely different language.
So, this is our third and final challenge. The language of DNA and RNA uses only 4 chemical letters. But the language of proteins, the molecules that do pretty much everything, uses a 20-letter alphabet made of amino acids. So how does the cell possibly translate from a simple 4-letter code into a complex 20-letter one?
The answer is ingenious. It's the codon. The cell doesn't read the mRNA message one letter at a time. Instead, it reads it in 3-letter words. Each one of these 3-letter codons points to one specific amino acid. So, for example, the codon AUG on the mRNA tells the ribosome, hey, start building right here, and the first piece you need is the amino acid methionine. It's the genetic code.
So this image here gives us the whole bird's eye view. This entire process starts with activating the raw building blocks, the amino acids. Then it moves into initiation, where the ribosome gets set up on the message. The main event, of course, is elongation, where the protein chain is actually built. It all comes to a halt at termination, and then finally, that brand new protein folds up into its own unique functional shape.
Let's zoom in on that start. During initiation, the ribosome grabs onto the mRNA strand and it scans along until it finds that AUG start codon. And then the key player arrives. It's a molecule called transfer RNA, or tRNA. You can think of each tRNA as a bilingual adapter. It carries a specific amino acid on one end, and on the other, it has a code that perfectly matches an mRNA codon. As soon as that first tRNA locks in, the factory is officially open for business.
And this right here, this is the moment of creation. The ribosome chugs along the mRNA one codon at a time. For every single codon, the correct tRNA shows up, bringing the next amino acid right into position. The ribosome then forges a powerful bond, linking the new amino acid to the growing chain. The old empty tRNA gets kicked out, and the whole process repeats over and over. It is a stunningly efficient assembly line, building a complex protein one piece at a time.
So, okay, we've gone from a coiled-up blueprint to a copy, to a message, to a final working protein. We've seen replication, transcription, and translation. But what's the big deal? Why does this tiny, microscopic 3-part system matter so much?
Well, the answer is actually pretty mind-blowing. This entire system, this exact genetic code, is basically universal. The same codons that tell your cells which amino acids to use also work in a bacterium, a mushroom, or a blue whale. This shared language is not only just powerful evidence that all life on Earth has a common ancestor, but it's what lets modern science do incredible things, like programming bacteria to produce human insulin for diabetics.
So let's do a super quick recap of the whole journey. Number 1, DNA is the stable master blueprint. Number 2, replication copies that whole blueprint for new cells. 3, transcription makes a temporary mRNA working copy. 4, translation reads that copy to build proteins. And 5, this whole elegant flow, DNA to RNA to protein, is the fundamental operating system for pretty much every living thing we know of.
This system is truly one of nature's most elegant solutions, perfected over billions of years. It's unbelievably accurate, but it's not absolutely perfect, which leaves us with one final and really fascinating question. What happens when there is a mistake, a typo in the code? Because that, my friends, is where things get really interesting. That's what leads to everything from genetic diseases to the very engine of evolution itself.